Imagine being confined in a cold metal cage, barely large enough to turn around. You don't understand the voices around you, and you can't predict when the next injection or chemical exposure will happen. You're poked, prodded, restrained, and eventually euthanized. Not because you did anything wrong, but because your body is being used as a stand-in for human biology. For more than 100 million animals in the US laboratories every year, this is the reality they live and die in. This isn't meant to shock anyone. It's not meant for us to pause and recognize how normalized this system has become, even though most of us would agree that animals are capable of fear, stress, and pain. And yet, despite all of this suffering, the more you look into animal testing, what animal testing event actually accomplishes, the more clear it becomes that it's not only not ethically questionable, but scientifically outdated. To start, the ethical concerns are significant. Animals in labs experience pain, stress, and invasive procedures, often without proper pain relief. Some are intentionally bred to develop painful diseases, while others undergo repeated toxicity tests that can cause burns, neurological damage, or internal organ failure. The public rarely sees this side of research, and since many species aren't covered under federal reporting laws, we often don't have the full picture, and the lack of transparency itself raises concerns. But ethics alone aren't enough to change scientific policy, and this is where scientific evidence becomes impossible to ignore. Numerous researchers have pointed out to limitations of animal models, but one of the clearest examples is in a 2013 study published in the Law of Proceedings of the National Academy of Sciences, which found that mouse models, the backbone of a lot of medical research, do not mimic human inflammatory responses. That means scientists can spend months or even years running experiments on these animals and still end up with results that fail when applied to human patients. The problem shows up in the drug development too. The FDA reports that approximately 90% of drugs that pass animal tests fail in human clinical trials. Some fail because they don't work in humans, but many fail because they produce dangerous side effects that weren't seen in the animals. When almost every drug collapses during animal testing, it becomes obvious animal data isn't a reliable predictor. Many people assume that it is. Another problem with relying on animal models is that they often miss conditions that are uniquely human. Animals don't experience certain disorders the way we do, for example. Complex neurological conditions such as many cancers, autoimmune disease, and even mental health disorders don't develop the same way in animal bodies. This means scientists end up studying simplified or artificial versions of human illness, which can only take us so far. When a model can't accurately replicate the condition we're trying to treat, the results become limited from the start. So the issue isn't just the animal testing sometimes fails, it's that many cases it was never capable of giving us the answers we needed in the first place. What's even more concerning is that these failures don't just waste money. They slow down the development of treatment that people desperately need. When scientists pursue a drug that appears safe in animals and spend years chasing what ends up to be a dead end, and while this is happening, patients are still waiting for effective therapies. The scientific community is starting to acknowledge this. The FDA and other agencies have promoted the adoption of new technologies that don't rely on animals. These include organ-on-a-chip systems that replicate the function of human organs, advanced computer simulations, and organoids grown from human stem cells. These systems often produce results faster and more accurately than traditional animal models. Another important point is that these technologies aren't just theoretical, they are already being used. For example, organ-on-a-chip devices have been used to model lung diseases, test how drugs move through the human body, and study cancer treatments. Human cell-based assays can screen thousands of chemicals in a fraction of the time animal techs take. And computational toxicology can predict how substances interact with human proteins in ways that animals never could. So what do we do? Even though we might not work in labs or write scientific policy, our choices genuinely matter. Supporting cruelty-free brands sends a financial message that consumers want modern, ethical science. Talking about these issues, whether in school, online, or with friends, can help raise awareness. And supporting legislation that funds alternatives to animal testing helps move the entire system towards better scientific methods. To summarize, animal testing causes intense suffering, provides unreliable scientific results, and slows down the development of a safer and more accurate research methods. We now have tools that didn't exist decades ago, tools that can mimic human biology far more precisely than animals ever could. If we wouldn't accept being trapped, restrained, or experimented on ourselves, especially for research that's unlikely to translate to human benefit, we shouldn't accept it for our animals either. And now that advanced alternatives exist, continuing to rely on outdated methods isn't just morally questionable, it's scientifically unnecessary and irresponsible.